So we had a few questions about TMT, and so I think Stephanie is going to jump back into that a little bit. And for those of you that are just joined, again, just a reminder about the Slack channel, and if you have any problems, say something in the chat. Otherwise, just you guys are doing great about keeping your comments and stuff in the channel. It's working really well.
Yeah, I appreciate that, because I'm hoping that that'll help us in the future. Okay, yeah, I did see a couple of just questions about TMT labeling. And so what we're doing here, it's kind of two purposes. One, it's able to run 10 samples at the same time through the instrument, so that saves you on a lot of mass spec time, ultimately helps with the cost. But it also gives you better quantitation, because now you've got all of those intensity values for all of your 10 samples within the same scan. And so you're eliminating some of that instrument variability from run to run, and you're eliminating that data dependent acquisition effects. So that is, if you were to do like the GLC approach that I talked about, where you just run a sample on a gel, you fractionate that, it's not labeled, it's label-free approach. Then when you submit those peptides into the mass spectrometer, every sample is going to be loaded into the mass spec individually, and each of the fractions of the gel fraction are going to be loaded individually. So then when it hits the that MS1 and it's looking for the most abundant peptides, that can be variable depending on what peptides elute from the column at that moment in time. So if you have a set of 10 abundant peaks that come off at the MS1, but then you've got a 5 other ones that are variable between elution profiles between your samples, you can get this smattering of missing values in all of those different samples. And so with the TMT approach, all of those hit that detector at the same time. You're selecting those same top 10, top 20 peaks for all 10 samples that are then going to go through the fragmentation process to give you the sequence. And then you get the quantitative information, the MS3 fragmentation. So your values, it cuts down on that missing value problem that you would have from a label-free experiment. The downside of that, if you have 2 different TMT batches, now if you have a high abundant peptide that comes off in these 10 samples in the first batch that it selects for the fragmentation for MS2, you'll get values in that first batch. But if another peptide is more abundant in the second batch, that peptide is no longer selected. And so now you have, instead of just 1 sample that has a missing value, now you have missing values in all 10 samples. And so that's why that batch effect between 2 different TMT batches is really significant, because you've compounded that now by 10 samples instead of just the 1. And you can really see that in your PCA plots. When you run a PCA plot, you're going to have the very first component is just separated by batch. And so there's some ways to deal with it, but it can lead to some false positives in your data or false negatives. So the tags themselves, they have that alkaline group. Let's see if I can get to the right thing to turn here. Where's my tag at? Let me find it. So right here. Yeah, we'll pull this one up. Switch my settings. So yeah, the tag is three parts to the tag. You have your mass reporter, which is where the MS3 intensities are going to come from, the mass normalizer to make sure this entire tag has the exact same molecular weight, and then the NH reactive group. So the reactive group is what's binding to the N-terminus tag, N-terminus of the peptide and lysines. So you'll have that modification in your database search in order to detect TMT peptides. This section here with the dotted line is that's where you can break apart the fragmentation, the reporter ion gets fragmented in that MS3 fragmentation process. And then each of the tags are separated by the charge. So you have a charge that is either on the mass normalizer section of this tag, and then you move a charge over to increase the molecular weight by 1 for each of those tags and separate those out. So it's based on the master charge values in order to get a separation of those intensities. And then so each of those will have their peaks in the MS3 scan. Hopefully that was helpful. If not, we'll answer more questions in the Slack. Okay, so that's data-dependent acquisition type run, where it really depends on the peptides that are coming off that column. The most abundant peptides are going to be selected for fragmentation, and that's going to be the values that we're going to get. That's where you can get a missing value for proteomics data. Doesn't mean it doesn't exist. It just means it was not detected in the mass spectrometer. So we have another newer approach is the data independent acquisition, which is not dependent on the abundance of those peptides. So when we have a data dependent, it's the DDA, data independent DIA. So for the one that I was just explaining with TMT, that's a data dependent acquisition. You have the MS1, which gives you the full peptide intensity value, and then it's fragmented into the MS2 to give you the sequence information. But each of those peaks in the MS1 are fragmented into a separate MS2 spectra. With the data independent acquisition, again, we're kind of it's all blending together into the MS2. So now the MS2 is consistent of all of those different MS1 peaks into one scan, and then we have to deconvolute that MS2 scan in order to get the sequencing information for all the peptides that are now present in that MS2 fragmentation. So the advantage of a data independent acquisition, instead of doing this punctate where you're just sequencing for the high abundant peptides that come off the instrument into the MS2 scans, you're now doing kind of a window-based approach. And you're trying to sequence everything within a narrow window of the retention time as they come off. You're really trying to sequence through everything and not just the top abundant peptides that happen to elute at that particular retention time. And Aaron Story in our group is on, and I'm sure he can answer a lot more of your questions about DIA in the Slack, because he's our DIA expert. So I am going to, any major questions about that, he's your guy. But with the data, I like the data analysis from a DIA experiment a lot better. It tends to have a nice quantitation data set. One of the challenges with DIA is we don't always get the depth from a DIA experiment that we would from a TMT. So kind of, again, depends on what your ultimate goal is. Do you really need to get more proteins identified? What are your sample? What's your experimental design to know what's going to be the best approach? I've got some notes in here. I'm not going to read this because you don't want me to. These are just in the slides, and I'll put this, it's in the Slack channel. And if you don't have access to it, let us know and we'll get it to you. But overall, for the DIA workflow, is some different parts to it. It's a little bit of a different process than our TMT experiments. So we have quantitative samples, and then you have to build a empirical library based off of the peptides that are found in those samples in order to do our database searching and identification of those peptides and sequences. So we actually, they do a really good job of trying to, utilizing the samples, making this pooled set of samples from all the samples you have, doing a gas phase fractionation using the DIA approach, and then building a chromatographic library in order to detect peptide sequences. And so using these gas phase fractionations, we can correct for when certain peptides elute at different retention times. You can correct the chromatographic libraries, the chromatograms, and correct peptide identifications. So again, I'm gonna let Aaron answer a lot of those questions. So we make the library, and this is really where where I pick it up is after Aaron has done all this work for me, making the libraries and doing the database searches, then I take it from the intensity data for the rest of the analysis. But the empirically corrected library accounts for using only the detected peptides for that set of samples, and you get correction for fragment ions and retention time. And then again, I just put some notes in here for your use later. Are there any questions about this part before I move on to normalization? We have
Do you
like—
mind elaborating a little more about when to use TMT and when to use DIA?
Yeah, so TMT, you're going to get more proteins identified. So if you have a very clear experimental design where you've got 5 samples versus 5 samples and it's just 2 sample group project, I would go TMT for sure. You'll get more proteins, deeper sequencing, and very quantitative data. If you have more than 2 groups and you need a lot more replicates and you've got to go across batch, then we may go back to DIA. Some of our serum projects actually work a lot better with DIA. I forget exactly. So for serum, we're getting rid of all the high abundant IgGs and albumin. So you do a different sample prep, and then we've found good success with sequencing those. Your proteome is a lot less in a serum sample than like a tissue sample where you're trying to get more. So we have had better success with DIA on those.
We had great success on serum samples with DIA as well. It worked really well.
Yeah, good to know. Serum, and then we started doing DIA with IP pull down experiments also, and that seems to work really well because again, you're not looking to have 10,000 proteins from a pull down. You hope you want to have that subset.
Have you used it for PTM analysis too?
We haven't done that yet, but that is something that we were looking at. So we've got a histone project where we've done our traditional histone PTM, but then we also want to do that with the DIA approach. So we may have that soon. So.
Great, thank you.
Yep. There's something else I was going to mention. I totally forgot it. It'll come later. So, anybody else? All right. So y'all be tired of hearing me by tomorrow, if not today already. Okay, so the next thing I was going to do is kind of launch into more of the pre-processing steps. And I'm kind of hoping this will be the rest of our afternoon. It's just how do we deal with the data? Got to swap. Okay, so like I said, we got multiple different methods, multiple different data that we're dealing with. I've got different database search algorithms, DIAs, the data independent, Mascot. I use Mascot a lot for histone PTM work. We used to do that also with IP experiments if you're trying to get count data. So Mascot will give you counts compared instead of an intensity that Max Quant provides. So the count data is literally just taking the MS2, the fragmentation spectra, and it counts how many times do I see the same peptide. Like how many of those MS2s do I have for a particular sequence becomes your spectral count. Those worked really well with pull downs in the past, and then there's a different normalization for count data that you have to do called NSAF, the Normalized Spectral Abundance Factor. And then that will account for the fact that you have longer proteins have the chance of having more peptides detected than shorter proteins. So you kind of have to normalize for those biases towards larger proteins. That's what the NSAP does. And Scaffold, in their analyzing in their software, they actually include the NSAF normalization function now. So it's actually in the software. Histone PTMs, I do have another tool that's in the in our GitHub for histones. And this is at the peptide level type analysis. So you're looking specifically at each peptide that's modified. So we have that new PTM Viz tool available for that. DIA, we're looking at the MS1 exclusive intensities. So the exclusive intensity is the abundance of that peptide as it comes off in the MS1. Exclusive, though, it actually is only looking at the unique peptides for a protein instead of the total. If you use the total, then you can get shared peptides across multiple proteins, and that could influence the data. So for those, we use the unique peptides, the exclusive intensities. Most of our stuff, we're using MaxQuant for the database search algorithm. And we have the label-free approach. So some projects lend better to a MS1 label-free. MaxQuant has an IBAC normalization function included in its software. And IBAC is, it's something absolute quantitation. I forgot, intensity-based absolute quantitation. So that's going to take the your intensities and normalize it for, again, kind of that theoretical number of peptides that can be identified for the protein. It's a similar type algorithm that NSAF does for counts, but this is on the intensity data. So you're just normalizing for larger proteins versus smaller proteins. And label frees are done at the MS1 intensity level as well. TMT, like I said, is done at the MS3 quantitation, looking at the quantitation of the tags. The phospho experiments. Phosphorylation experiments, we do 2 different analyses, and those are all TMT. So you take the protein lysate for the total proteome. We do the full TMT workup of that protein analysis, but then we do an enrichment for phosphorylated peptides to do the phosphorylation analysis, and that's done at the peptide level. And I'll talk more about that tomorrow. And again, we have the ProteoViz tool for phosphorylation projects that's available on the GitHub. Okay, so this is just kind of an architecture of the workflow of how things working. We're currently working with Google right now to develop some of this stuff in the cloud, and which I hope will be a nice resource for all of you to use later. Just to let you know that is coming. So we have take our raw data. We got either a mascot or MaxQuant database search. Then we can dump those results into Scaffold. Scaffold is a nice viewer software to look at the more details of the data. This is really important for publications. So I found that it's easier to submit data that you need to the PRIDE repository by using the Scaffold tool. Scaffold will export the MZI-DENT files that are necessary for a PRIDE submission, and it makes that process a lot smoother. So I use that a lot for PRIDE. And then for DIA experiments, we're actually using the exclusive intensities from the Scaffold to import into R to do the downstream analyses. So most of all the statistical analysis stuff is actually done in an R platform. And then we're trying to build out reports and kind of summaries of what the data looks like to help end users understand their data. So this is kind of a workflow that I'm currently working on with the Google team in order to build cloud computing and have some more training modules and stuff too. So NIH really wants to establish this training platform for various omics data sets. And we're kind of working on a proteomics one. So if you have some ideas or anything you want to see in a training, let me know. So ProteinOrm is a tool that we developed in order to evaluate normalization methods. because as a service, when you're running lots and lots of projects, each protein distribution is going to be different depending on the context and everything. So instead of just like choosing my favorite normalization algorithm that I really like, I was trying to build a tool to evaluate those metrics and see how the distribution, what's the variance look like, what's the intergroup correlation values, and see how the distribution is changing with these methods. This is still, there's a lot of work we could do in proteomics to improve things. So I'm definitely open to ideas, suggestions. Anybody wants to collaborate, that would be awesome. Okay, so this is our app, and I have a little demonstration I'll show here in a second, but it's just an R Shiny app that we pop up, and we've got two different inputs, the peptides and then the protein groups. So the output from XQuant gives you two files, one peptides.txt and another is a protein groups.txt. So the peptides file is going to give you all of your intensity values based on each individual peptide that's identified. The protein groups actually takes those peptides for the same protein and sums them and gives you a summed intensity of all the peptides identified. Normally, for most experiments, we can start with a protein group file, do all our analysis, and have good data at the end, find some nice differentially expressed proteins that makes biological sense. You can validate them, write a paper, you've got something to go with. Sometimes there are certain proteins that are really biologically interesting, and if they're lower abundant, one bad peptide identification can skew the protein group intensities. So for some of those, we have to investigate a little bit deeper at that peptide level in order to do some corrections of the identifications. Because you're relying on this database search algorithm to take that spectra of those peaks and then match that to the theoretical peptides in a database in a FASTA file. And it's not always, there can be some errors in that. So we have this other way of filtering peptides where you can actually start with the peptide file, use the top three method, which will take the top three peptides for that protein and sum those together to give you a new protein group instead of using all the proteins identified. So there's just some flexibility there. It's one of those options and one of those things that when you're really diving into a specific project, something to keep in mind. That you sometimes, if you really want to spend a lot of money to validate a certain protein, you might want to go back and look at those specific peptides and make sure it looks good before you spend a lot of time on it. So we got the peptide side and the protein side. I think what I'm going to do now is actually see if I can switch to my other computer and show you what it looks like. If I do it on this computer, it will crash my Zoom. So I tend to have to try to remote in. Well, come on. Let's see if it'll work here. I was having this issue earlier. I may have to do it later. Let's find out.
Stephanie, do you need me to call IT?
Yes, now I think it's working now.
I was just kidding. If your campus is like ours, that's the last. Never mind. I'm sorry.
I'm like on a first name basis with IT. It's fine. Okay. I think I may have to rerun this because otherwise my fonts and things are not gonna look very good. So bear with me just a moment. I am gonna save that so I don't have to retype it. This is what you get with real live demonstrations. It's like, hang on a second.
The questions that have been coming up is about quantification in DIA and how that's done, and that's what people are
talking about.
Yeah, Aaron's doing a good job of answering that, and so is Julie.
Good deal. That's what I got y'all here for.
That's right.
Don't save. Yeah, so I've got this example data set that we're gonna go through. One, we did the same samples with the TMT and DIA, and so that we can do that comparison of what that looks like. So this little app here is my Proteonorm app. I'm gonna run it.
What organism were those samples?
It's fish oil. I think it was human.
Okay.
Don't quote me.
It's a mouse. It's a mouse model,
Mouse.
actually.
Okay. It was mouse, human, rat.
Yeah, one of those, right?
Yeah.
Whatever.
Will you talk about the importance of database matching too? Like how to create the database a little bit more and go into that and using CRAP versus not using the CRAP file?
Yeah, I wasn't planning on, but we can
Oh.
talk about that a little bit. Yeah, database is that's always a fun challenge. Okay, here I'm in my Proteonorm app, so I'm just going to browse for my protein file. So I'm in our Kinter TMT data set. Gonna go to my data. So here I've got a peptides.txt and a protein groups.txt. These are the output files from MaxQuant. So we're gonna load in our protein groups. And what Proteonorm does is it automatically detects if you have TMT data or if you have a label-free experiment. So it's going to automatically detect the structure within your input file. And what I'm looking for, MaxQuant has a reporter intensity corrected, followed by a number, followed by whatever label you labeled it when you set up MaxQuant. And so this tool is looking for this specific reporter intensity corrected, and then it finds the numbers. So we have 11, the TMT 11-plex detected in this particular experiment. The corrected has to do with the fact that you can have a little bit of shift in the spectra for the TMT reporter ions. And so every single TMT kit that Thermo provides, they have a error correction sheet associated with that lot number. And so the lot number of your TMT labels is really important. Then I can import that correction table into the mass spectra, into the database search, and it'll correct those values. So that is done for if you don't have, and you're just trying to make it populate a file and you didn't use TMT, you got a different structure, you can just label each of your columns of intensities of your samples as intensity followed by like a 0.1, intensity 0.2, something like that. It's just going to look for that intensity. And all of that description is there in the GitHub. So then I have, I really should turn off my slide. We won't be here long. So I'm going to just populate my names and my group, and then I also have a batch function. Yeah, my things are always full. So we've got all of the different samples, and this particular example is the same samples, 11 through 15 for the Q versus 71 through 75. Those are the two different groups, your control diet versus the fish oil diet. And then it's the exact same sample in the second batch. So all of these samples are done in triplicate, but across batch in order to really test the batching effect with the same samples. That makes sense. So all those 10 samples are pulled together in TMT1, TMT2, TMT3. Then we also have a pooled sample. The pooled sample is the pool of all of those samples in those different channels. So that's all of our data. Then we go to filters. So here you can kind of get a look at the overall distribution of the intensities of each of the samples. This is just raw MS3 intensities, and it's color-coded by the group that we selected. And you can see here, for some reason, that third batch actually had overall less intensity than the other two batches, for whatever reason. Then we also have options where I can get rid of the pooled samples if I want to. So any outlier sample I want to remove, I can do that here. Then we can look at our normalization methods. It's going to take it a second because it's all running in the background. Again, so this is just, we have 8 different normalization methods that came, we didn't create these methods. These were from the Normalizer DE package plus a couple other packages. So this is really just an evaluation of those methods. And you can see that overall batch 3 has lower intensities than the others. So let's go to.
Hey, Stephanie, can I jump in here for just a second? While you're talking about normalization, there's a question that came up here from Gazelle. It says, can we compare the protein intensities coming from 2 different instruments? Are there special normalization techniques to do that, or do we always take the intensities coming from the same experiment only?
Yeah, if you're trying to go across experiments or across instruments, you're definitely going to have an instrument batch effect. And so you can kind of do that, and got to remember that this is a discovery phase type analyses, and you want to validate those findings later. But you could do Lima model has a batch correction in their statistical design. So there's a few different ways to do that. And I don't know that I have like the best answer for all of the normalization stuff. So I'm going to kind of walk through what we do with this tool, and then really hoping we can have that open discussion here this afternoon. Those are exact some things that I want to talk through. So I think I'm going to answer your question here in a little bit, if that's okay.
That's right. Aaron also put a reference in the channel, so we got some information. Good deal.
There's a lot of things to look at for that and consider when you're doing those type of approaches. So we got, we're looking at our coefficient of variation. For a coefficient of variation, you want that to be low in the method that we want to choose. You've got the mean, median, absolute deviation. Again, you want those values to be low, and then the pooled estimate of variance. So all 3 of those are really looking at the variability of your data, and you want the variability to be low. The intergroup correlation is looking at how well your replicates within the same group are correlated. So the correlation should be high, but the variability should be low. The one that really helps the most a lot of the times is looking at the log-2 ratio. So this is taking kind of that overall distributions and the ratios of your control to your treatment, and seeing how does that impact your fold change. So the overall assumption for all this stuff is that most of the proteins are not differentiated. You're assuming the majority of the proteins are not changing in the analysis, and you're looking for those few that are changing and are significant. And a lot of that's going to be kind of, do we know anything about the effect size that we're looking for in your project? Do you expect a lot of changes or very few changes? So Cyclical S and VSN tend to be the better performing methods for most of the projects we see. VSN tends to work better with those lower abundant molecules like the phosphorylation PTMs. I have noticed that. But ideally, the better we do in the sample prep side, the better the samples are that go through the process. Ideally, all of those metrics will be very similar, and it won't impact the end results that much. The missing value one is the one that you can really see in the TMT batches. It really pops out here. So all of these are now missing values for those proteins within this batch that were detected in the other batches. So this is what impacts our PCA charts and the separation. And if I actually remove that 3rd batch, you'll really see that PC1 separate between the first 2 TMT batches. I meant to add in a function here where I could just do this like by group, but I'll do that later. I don't really use the app much anymore. I do it actually in the R code. I'll take it a sec. So yeah, if you're just looking at 2 batches, you can see 80% of our PC1 is really separated by batch. And you can actually group that, color it by the batch. And so they're very obvious that how the TMT really impacts your batching. One other method that I've used before in the past that is you can use a combat function for microarrays to try to break those batching effects. Again, it's kind of an older method, and which one's best is hard to determine when you don't have a standard in the experiment. So, but what you would do with the combat is you take those 2 different batches and you have to normalize those separately, and they have to be normalized. And then you could do the combat approach to break that batch. And if I do that and I show PCA, then you actually can start seeing the batching effect break. The other way that we can do it is normalize it here with like Cyclical S, one of these methods, get those normalized values, and then we actually put that batch into the Lima model when you're doing the statistics. And now it's taking an account into the statistical model, but you won't see that in the PCA. It's actually done on the statistical side. So there's a couple options that we've utilized. But I really tried not to do experiments with a lot of TMT batches. If you can just avoid that, that'd be better. Okay, anything else I want to show on this part? Oh, the DA test. So we did add in this functionality also. This is where you would select whatever method you want, save that file. Now you have a normalized protein group file for your data. We do have, we also included the DA test from this Russell, somebody, I forgot his last name, but he has a GitHub for the DA test, which is kind of nice to test all of these different statistical methods. And so it'll take your normalized data. For that test to work, you do have to impute the missing values. That's the only reason I have this imputation function here, is in order for the DA test to work. Because like t-test and some of those require you to have, you can't have any missing values in their data. But it's kind of a nice way of testing all of these different techniques on the data, and it'll provide power calculations and effect size information and help you to see which of these statistical methods may apply better to your data set. Most of the time, the log Lima ones pop up as one of the top methods. And so we tend to use a lot of the moderated t-tests with the Lima package for the statistical analysis. Okay, let's go back to, my Zoom is like interfering with my ability to get to my remote. All right. But normalization is a big deal with any data set, any omics data, right? So once we have normalized data, we do have for the output these normalized, try to make the interactive plots for your data distribution. I'll show a lot of that as well. So pretty norm, we've got the 8 different methods: log2, transformation, median, mean, variance stabilizing normalization, quantile, cyclical S, the robust linear regression, and global intensity. Trying to just kind of put some notes in here, so hopefully we can not bore you too much with me just reading them.
Steph, we got a couple of questions here. Talking about normalizer DE.
Yep.
Sometimes see most of the normalization methods return similar results, except for the one that's drastically lower, which always makes me suspicious. Could this be an artifact, or do some methods just work much better?
Yeah, it really is going to depend on your distribution. Like the one that, the variance stabilizing normalization method I've seen be drastically different from some of the others, but that's usually going to be in one of my data sets where I have really low abundant peptides and proteins. So like the PTM level, where it's those low abundant ones, tend to be affected by more variance, and then the variant VSN tends to handle that better. I actually did one of our phospho projects just before this. I ran all 8 of these methods on it, to then see how the differential expression changes at the end and what drastic differences are we seeing by doing a different method. All of your high abundant proteins tend to all be significant. They've all popped out. The ones that are changing is the lower abundant one where you may have some missing data included in some of the peptides, or could be a bad peptide identified at one of those lower ones. So I think it really kind of depends on your dynamic range of your proteins and your distribution, how much variance you have. It's all going to be different depending on different data sets. This is why eventually I'd really like to have a standard. I'd really like to have a standard in the data sets that I know what my ground truth is and that we can normalize with that. But that's what I would like. So in the meantime, I'm just trying to kind of look at what's the overall structure of this data? How are they correlated? What's the variability in the data sets? And then at the end, do we see those proteins, do they make sense? Can you put the data that we see with other data that the researcher has? Is it making biological sense? And then kind of having to validate some of those.
Can you do more than 2 groups in proteome?
Yep, we do. I'm not sure if it'll do more than 3, because I had my postdoc develop it, and so I had 3 groups and stuff, and we had to go in and do some changing of the algorithm stuff in order to do 3. And then I think I broke it like 100 times before we got things to work. Always a work in progress. Cyclical S is the one that is from the Lima package, and it was really designed for microarray. A lot of the stuff we use is designed for microarray batches. And so you're trying to fit this lowest smooth across each pair of chips in a microarray experiment. Well, in this case, it'd be kind of like going across batches of TMTs or different runs of samples through the mass spectrometer. And so it's trying to it's going through this curved data distribution between your M versus the A, the mean versus the average, and then looking for, we'll just go here to show it. So you have your MA plot and you've got this curved kind of distribution, and really the assumption is that there should be no change and they should be centered around 0. So you try to fit this curved line through all those data points to normalize it to become a flat distribution. And then you see all the spiked in proteins that they have in this example actually show up as the ones that are differentially expressed in the group. So it's cyclically applying this kind of linear function onto a curved graph and flattening that out. Other people could probably explain these algorithms a lot better than I can, especially right now, but there it is. But yeah, VSN I've seen a lot better with your lower abundant stuff that has a lot more variability in the data. Let's see if I can show you an example. I'm going to cut through here. While I'm on it, I do have that example from this one. So this is a data set that we just got published like 2 days ago for a phosphorylation experiment looking at cells treated with the vincristine. So these were the different metrics that we had at the protein level analyses, and you can see most of those algorithms really performed similarly. There wasn't much difference in the different algorithms. You kind of see their log-2 ratio is really nice. We do have kind of this, we have 2 different batches of TMT. So you can see a strong batching effect between those 2. The group that are really focused in on is this VCR versus DMSO control. And so we're really, you can filter out the batch 2 to just have batch 1 and do the analysis within that batch and that'll also kind of get rid of the batching effect. Now, if you're trying to compare this sample to this sample, that's when you got to deal with batching effects. And really, it's a problem. It just is. I don't know that there's a good solid solution. Some people I've seen try to do like 2 or 3 different normalization methods in order to break the batching, but then you kind of almost over normalize your data at that point. So here, just try to kind of do the best we can and get to the biological outcome and help the biology help us find the interesting proteins. So if I did this with all the different methods that we have at the protein level, most of them kind of give me the same similar results. Really, June is the top protein that was significant in this data, which was perfect because this was really more looking at phosphorylation, not so much changes at protein levels. But if we do the mean or median, then it picked up a couple, this other protein as significant, but they're very similar in distribution and range. It's just really small changes within the threshold of significance that you're setting. I think it was like 0.99 compared to like 1 or something like that. So it wasn't changed drastically, which makes me feel a little bit better about my normalization stuff. And it's similar for the other comparison. The phosphorylation part, you start seeing some spread and differences and variability among those different methods. So it's starting to kind of give you a little bit different distribution, again, because that's going to be your enriched phosphopeptides that are those lower abundant molecules, and they're going to have more variability in the data. So then when we look at this plot, you can start to see Cyclical S kind of separate out from the others. And I think that's the one that I actually used for this analysis. And if you look at your missing values within one batch, you don't have that many proteins that have missing values within a single batch. It's when you add in multiple batches that you really start to have bigger blocks of proteins that are going to have a missing value and kind of give you a big batch problem. So this is going to be at the peptide level. And you can see, again, it's like each method really kind of gave me very similar results. There wasn't a whole lot of change, just a few molecules that changed, and they were pretty close. It's just where do you set that significance threshold that really made the difference. So I kind of looked into one of the lower abundant molecules with one of our tools and try to see, this is the CFP36 peptide. This is the peptide sequence that has got a phosphorylation on S192 of that particular protein. And so if we look at an MD plot, the average expression of all those intensities on your x-axis versus the log2 ratio on the y-axis, this protein is kind of one of those more lower abundant proteins. So it's not really significant in the Cyclical S with the p-value just misses 0.05, it's 0.056. And then if we look at global intensity, it's now significant with your p-value is 0.0487. Very, very similar. So that's just kind of a parsing of where do you set your significance of your p-value. So each of those methods on this particular data set, it really performed very similarly. There was just like a handful of proteins that were not identified the same. So kind of doing some of those checks made me feel a little bit better. So this is the example data set from the fish oil experiment. I guess I should do this. So for this, like I said, in the TMT batch, we had control diet, 5 samples, fish oil, 5 samples, and repeated those for 3 batches. So it's the same samples in batch 1 or now batch 2 and batch 3. They're just chemically labeled with different TMT reporter ions and ran through the instrument 3 different times. We did the same samples in triplicate as a DIA project. So now you've got the control one, number 11, 3 times, and then 12 3 times. So all of those are just run through a DIA experiment instead of multiplex together. So if you look at the TMT, again, this is what I was showing in the app. You've got those batching effects between the TMT batches. And you can see clustering, separation of batches, not by groups, but they're actually separated by the batch. We've got about 5,000 proteins identified, 128 were contaminants. The contaminants is coming from the database search. So the database search, you have a FASTA file that has all the sequences for all your proteins of a particular organism. And then you have a secondary database that's a contaminants database that includes things like keratin, any known contaminants that you can get just through the sample prep process. So like keratin comes off of your skin when it's in the lab, it's in the air. A lot of those are included in the contaminants database. And MaxQuant will flag those protein IDs with CON so that you know it came from either your real database or the contaminant database. The algorithm also does a search with the reverse of all of those sequences. So it'll use the reverse sequence in order to determine the false discovery rate of identification. So anything that hits to the reverse sequence will be flagged with the REV at the very beginning of that sequence ID, the protein ID. So those are ones that we want to filter out of our data at the very beginning, and we don't analyze those particular proteins. So that's where this number of protein contaminants, we got rid of those proteins in the analysis. So then you're left with 4,347 actual proteins in the data set and about 49,000 peptides that were used to make the protein group file. There's a lot of information here, so sorry if I just like forget something. Sorry.
That's okay. We got a question come in, so take
Yeah.
a deep breath here. I'll read it. So the question is the Cyclical S the most popular normalization method? How do we know which one is the best when you're trying to decide from 10 different methods?
Yeah, so Cyclical S is kind of my default. If they all look the exact same, I default to Cyclical S. It's part of the Lima package, and we're running the Lima algorithm. So yeah, it's always a good question. I don't know that I have like detailed specific answers, but that's what I do. I kind of default to the Cyclical S. The one that you'll be able to tell a big difference if VSN is performing better. RLR has actually performed better on some of the data sets as well, but I'm not as familiar with that one, but it has given me some better metrics. And the metrics I'm looking at are those in the proteonorm with your, you want your variance of those box plots to be the lowest and the correlation to be the highest. And the log2 ratio is the one that really kind of helps narrow between multiple ones, because you can really see the shift in the distribution. And you want that distribution to be centered at 0.
Okay,
Okay.
Steph, you need to be thinking about the next break here coming up. So
Sounds good. Let's get through this Kinter part, and then let's take a break at that point, because I
Okay.
think I'm just about done with this.
Okay, that sounds good.
Thank you. I appreciate that. Okay, DIA, same samples. Now we're just running through DIA, and you can clearly see the difference here in our missing value chart. We don't have we don't have those big blocks of missing values in that batching effect like we did with the others. So that is a huge benefit of the DIA. However, instead of having 4,600 or however many we have, we now only have in like 2,700, 2,600 proteins identified. So you don't get the depth, but you get less of the difficulties in the analysis. And you can see in the clustering, you actually are starting to see a separation between your biological groups and not batch. So it's actually starting to separate by the biological conditions. So that's super helpful. And then we got all the differential. This is just your which ones were significantly upregulated versus downregulated. And then what it did is did a comparison of the TMT DIA significant proteins. How many were significant by both methods and how many of those are the same in both methods? So TMT, we had 4,300 total proteins, 127 of those were significant, giving you 3% significant proteins. DIA, we had about almost half total proteins identified, and more of those were significantly different with 5% significant. So if you look at the upregulated, significantly upregulated significant list, only 39 out of those were the same in both the TMT and the DIA experiment with the same samples. You do have some unique ones that are associated with either method. And the same similar results with the downregulated significant list.
Can I ask a quick question? How much was the overlap between the two and just identified proteins? Like if you go one slide back, like were the 46 in the TMT up even identified in the DIA, or were there not even in that data set? Like how overlapping were they in general?
I do not remember that.
Okay.
We can look at that before tomorrow.
Another question I had, sorry, because you were pointing out that TMT has much more missing values than DIA.
Yep.
So overall, there are less identified significant proteins. Is that due because we're dealing with more missing values and it's not a parsed data set, but it's more parsed than the DIA.
Right.
So that account for that difference, or?
Yeah, I think so. Because I think you're going to have more false hits with the TMT due to the batching effect and stuff. So that's where I went to this next slide here. You can actually see the full proteins from TMT versus DIA. And I try to kind of flag out what proteins are the same in the two experiments and where they fall in the significance. And so the top abundant ones are really, they're all in the same kind of quadrant up here that I have all highlighted. There's not that much difference on this level of proteins. It's the ones that we've identified that you're missing in the DIA that we don't have here. And then some of these that are flagged as significant, I think could actually just be due to the missing value problem. So this is where, let's see if I can bring it up. Hopefully I have it on this one. I've got data everywhere. Let's go to this one. So this, these interactive plots is where you can really start asking questions like that. So you've got all these that are down here. Let's go to the volcano one. Come on. Computer doesn't always like to do all of this and, oops, wrong one. It's not what I wanted. We have MD plots and we have volcano plots. So both are super helpful. And then we have them with the no adjusted p-value versus the adjusted p-value. So you can look at your differences in how the adjustment is actually affecting things. But a lot of these here on the TMT, this is where you can start looking at these outliers and see that those samples actually have a missing value. And so your overall fold change is actually not that different, but it's got a missing value here that's influencing that value. And you can start to see here within these replicates, this is batch 1, this is batch 2 and 3. So you can actually see in the normalized data set kind of that batching effect and how it's impacting those results a little bit. So I often use these interactive tools to really try to get at the individual sample information to help make a determination, is this a strong difference or do we have some variability in those groups and those sample replicates that may be influencing the impact of that fold change and that significance. So like this one is a very strong difference and all the samples are really tightly similar. And so this is really, if I'm going to try to find my top list that I want to go after, kind of diving into that deeper approach of what my different replicates are doing has been super helpful.
Yeah, this is super helpful. Thank you for elaborating.
Yeah, no, this is probably what we do in most of the consult meetings at the end of a project is spend some time here and just showing you like how do you interact with this data, find out more of those details so that we can ask those very things. How much confidence do we have in our fold change and our p-value? And then this, just to wrap up with this part real fast, we've got this, we also did a targeted approach for a few of these proteins. And you can see that the targeted approach with the catalase is higher in the official compared to the control, and it's the same in the TMT, and it's the same in the DIA. So those that are like really popping out biologically tend to come out in both the TMT, the DIA method, whether you have the batching or the not, and those different normalizations. Still, the most biologically interesting ones seem to pop out into the top of that differential list. So that always makes me feel better. It's when you're trying to get to those ones that are kind of there on the edge of your significance, you really want to kind of take a little more time with, look into the peptides. What did we actually identify? That's where it can start becoming time consuming, and you really need to know more of what your biological story is. Okay, that kind of brings us up to closer to getting to more discussion stuff. So